I'm Eve Schneider, and I'm an assistant professor in the biology department at the University of Kentucky. So, my lab's primary research interest is the development and evolution of the sense of touch. So, right now, my lab is interested in understanding differences in how the nervous system is organized in species that really use the sense of touch every day to find food, like the ducks you see dabbling here, and species that are not tactile foragers. So, one of the things that makes the duck a great model organism for studying touch is that the skin of the bill is packed with corpuscles and sensory neurons of a similar type and density as is found in the human fingertip. So, we can do all our experiments in duck embryos before they hatch because many of the components of the touch system have developed by the time of hatching. So we use a variety of techniques, including patch clamp electrophysiology, as shown here, evoking mechanosensitive currents from a sensory neuron. And recently, probably the most exciting thing in my lab is I built a 2-photon microscope that relies on a high-powered infrared laser. So because it has a longer wavelength than visible light, this allows you to penetrate very deeply in neural tissue with minimal scatter or bleaching. So we can use this to image neural pathways labeled by viruses that cross between synapses or with live cell calcium imaging. So in terms of communicating with my grad students, we have lab meetings every couple weeks. We use Trello a lot for communicating the kind of nuts and bolts of projects, and lots of informal meetings. And in the beginning, I do a lot of hands-on training for learning how to do experiments. There's a lot of kind of organic, unforced socializing that goes on in this department. Nothing really. I just try to give them enough time outside of the lab so that they can do whatever they want for fun, and I don't need to know about it.